Okay, so this is our database. There's a link at the top of our Moodle page. The last link is a link to this as a Google Sheet. And so feel free to go and double check that your data has been entered, because this is essentially your participation and your coursework is being captured within this. So
And can I
Matt,
can I just add one thing, Adrian, quickly? I'm sorry to interrupt.
no, of course.
It was just, yeah, so just on that note, if you would double check that it's in, because I had some trouble, there's some duplicate. Each of you uploaded oftentimes like your group information, but then other times you uploaded just your individual information. So there may be duplicate records. I tried to sort and avoid that, but you'll see under the team, sometimes there's individual names, sometimes there's a team name. So I did the best I could, but there still needs to be filtered a little bit. So just have a look through. I graded everybody accordingly, and I do have a couple missing files. Of note, if you could upload your files, like not in a PDF, but in Excel or a CSV, that would be easier for me to extract it and then put it in here. Because the PDF files, I have to go and manually enter all those observations, which I don't want to do. And
No, don't
then
do that.
and then one student just sent a file that it was incompatible. It didn't open. I'm not quite sure what it was. It wasn't text. It wasn't CSV. So just consider what you upload it in. Excel is the easiest, but anything in database form.
Yeah, please, the instructions were very specific on this to make sure that you download it from Google in either the Excel or in a TSV or a SCVS or TSV, whatever, text or text delimited file or comma delimited file or tab delimited file. I don't care which one, but just something that's text-based. Otherwise, we really do. And I think that, Matt, you're having a problem with the Apple program, which is not, it's not bringing my I'm not remembering the name of it, but that's always been a problem for me as well. And
Okay.
I think you can open it up somehow, but I don't remember how.
Okay. I'll poke around with it.
But yeah, just try and do that. It makes things a lot easier on our end. So double check this to make sure it's great, works right. But this is how I want. This is how I want you guys, this is the data that you're going to use for your group presentation, okay? So what you're going to do is you're going to focus on the part of the data set that you were responsible for in the electroshocking period, okay? So remember, that was the first day that we got you out into groups. So get together with your group. I will leave time in next week's class for you to do that. If you want to try and keep it in class, extract the data, make a couple of slides with with figures in them that show the fish that you caught, and make some comparisons to what was caught in other locations. And then you can just what we usually have is a little section where you talk about some of the species that you had and some of their characteristics and some of their habitat. The reason why I wanted to direct you to FishBase was so you had a resource that can help fill in a little bit of missing information. Try and make it interesting, but don't make it too long, okay? We just seriously, I don't want you guys to spend, 15 minutes would be the absolute max on one of these presentations. But if anybody knows how fast time goes and how long I can end up talking, it's me. So, and my apologies for that. Okay, so I am going to only do two things really quickly here that I wanted you to be aware of. And then I will, why is that doing that? I have a chat. Yeah, 10 to 15 minute presentations. And that would be, I think, like as long as ever needed. I mean, in many years, I would say that 7 minutes is probably right around where people are, just because 5 minutes is so short that, it's like, it's hardly enough time to say anything. But I want everybody in your group to speak. So try and divvy it up, as you talk about things, and feel free, like if you only had a handful of fish, to broaden it to talk about some other species at other locations that weren't present in your location, and maybe talk about why you think that is. But I'll give you an example, a couple of example presentations so you have something to go by on that. And then feel free to become armed with questions for next week. Okay, so the things I wanted to talk to you about today were very briefly that there are a couple of really important things in Excel that I think are really useful for people. So for the first one is filtering. So if you click at the top here, and I know some of you are going to be like, I know this, but I just need everybody to hear this first. All right, so if you go to the top here and you go to data, you have this opportunity to hit filter, which allows you to get these little arrows at the top so you can pick your team, for example. So let's say you're like, I only want to know what bigger, badder, beta, and than ever. You guys got to figure out which one of these you're going by. Then you can choose your team, and then only the data is available for your team. Okay, so this would be one way to extract information about your species. Okay. Now, another way, which is actually probably better, I'm going to turn off this filter by just clicking on it again, is to make a query. Okay, and so if you're not familiar with queries, you insert these pivot tables here, and these pivot tables allow you to, and I'm going to make it in a new worksheet. You get this. Of course, this thing's in the way, so I can't use my. Okay, so in a pivot table, you have this ability to turn your data from a whole sequence of rows into a mixture of columns and rows. Okay, so for this case, let's say I want to know let's say I want to know the species code. So I'm going to put the species code in columns, and I'm going to put the team names. I'm just going to make this up for right now. I'm going to put the team names in the rows, and I'm going to make I'm going to take the number of fish into this values, and then if you got to go here and say it's the sum. All right, so now you would have the sum of every fish caught in each of the different groups. This isn't going to work quite well because I haven't gone through and made sure this is super cleaned up. Okay, so this type of thing also then points out where you may have some issues with some of your numbers and stuff. But this is a way, for example, to get information. I'm going to change that from group to location. Okay, so now we have our locations, and we have all of the fish that were caught. All right, so what you can see now is you have a table that you can work with, and you can also use, let's say now you want to know a specific group. You can then sort of take out group, and you can put it in your filter, and then at the top you have a filter here that allows you to choose either Billy or Eliza. All right, and I don't know why those are the only two groups available, but that's what it is. Okay, so I know why, because it's your team.
Because it's the team.
All right, so then you could look through and you can get everybody within each location. All right, so this can get away from you, and I don't want to spend any more time on it, only to show you just these couple of things that will be really helpful as you try to get your data organized. This is a terrible example though with this team now all over here. If you put team at the top in this filters though, you can actually just like click on one of them, and then you'll only get the data for that one group. Okay, so this just allows you to quickly extract data that then you can put into tables or into bar charts to sort of inform or pie charts to inform the questions that you're really interested in, which is like, what are the species that were found at my location? And that can be for your group project or for your individual project once we get you that data. Okay, so that's all that I have really on this database. I will stick around, probably not this week because we're going to run out of time, but next week, and I will make sure to allow myself some availability for you. And please reach out to myself and Matt if you have questions and haven't used Excel a lot and aren't comfortable with the kind of extraction of data, and we'll work through it together and make sure that you are in control of your data. Okay, so that's that. Any questions? All right. And I'm going to wrap today with a story. And I would push this off to next week, but my experience is that I will run out of time next week if I keep pushing off things to next week. So I'm just going to power through this. That is, if I can see it. Where's the PowerPoint? Too many things open. Okay, of mice and men, no, of fish sampling. So I'm actually quite uncomfortable talking a lot about my own research, but I think that this experience that I had really speaks a lot to my time as both a graduate student and basically just out of undergraduate. So this is me when I was much closer to your age, and I think a little bit more relatable in terms of like trying to understand, because now the stuff I do is like I never get on the field. Ask Matt; he's like the guy shows up sometimes, complains about pulling fish, and then goes back saying his back hurts. Back in the day, I used to pull this stuff and do more work actually in the field. And so this allows me, I knew you'd at least show up again when I said that.
You're very helpful in the field.
He's just saying that. He's just saying that. I feel like I get in the way more now than I used to. All right, so what I wanted you to realize by first introducing you to the fish base was that there are tools out there, okay? And so before you start anything, fish base is really helpful once you know what fish are there, but there are some things you can do that don't even involve understanding what fish are there, okay? So for example, you can get information about the oceanography. So these are satellite images that are now being transformed into data that you can use to really understand what's going on. So this is the Gulf of Maine, and one of the things that I'm going to talk about a little bit today is the oceanographic structuring that occurs in the Gulf of Maine associated with this eastern Maine coastal current and this stratification and warm surface waters that occur in the western part of the Gulf of Maine. So this kind of information is really useful if you can get it. So sometimes you get it as part of your bottom trawl data. It actually has a bottom temperature, which is great. But other times you got to extract it from either a satellite derived product or something else. Just some really amazing images showing, in this case, the Gulf Stream with its Gulf core rings associated with complex oceanographic processes, but these can be associated, and during my time in Long Island, this could sometimes get really close, and all of a sudden we would have tropical fish at the piers, in particular lionfish would show up at the piers on Long Island shores. So knowing something about the oceanography is really helpful, and there's a lot of satellite-derived products that we've started using quite a lot. There's also a lot of bathymetry data. So this is an image showing Georges Bank. So we would expect the stuff that is found on Georges Bank is going to be different than the fish that are found in the deep waters here, but also in these deeper basins in the interior Gulf of Maine, which are actually very, very thick with sediment. Whereas Georges Bank is mostly turns into a sandstorm every tide as those currents really start ripping through there. Again, information that you can get. You can get information about what's happening dynamically. So obviously you're going to have very different answers to what you caught if you caught if you sample during low tides and high tides. In this case, you'd be doing a tide pool study in one, and you'd be doing trawls in the other. And so this information is often available through databases that are available online. There's a bunch of them, and there's also some R script now that can allow you to identify the tides at the specific location that you're interested in. That's not the only thing, though. We get precipitation data that is available both as a product through NOAA and the Climatics Data Center, but also through gauges and other methods to understand sort of stream flows and how they're influencing those organisms. You get anomalies, how much they're different this year compared to other years. Is this a dry year? Is this a warm year? Is this and that helps you understand your data from that year. All data that you can collect without going into the water and actually catching the fish. And certainly after having caught your fish, you should be trying to get this information to sort of understand what those processes are. That's not the only thing, though. Here's the gauge data. So here we can actually figure out what the relative amount of rain is in a certain year. Okay, so this is just an example sort of showing the average data, but we can get daily data, in fact, sub-daily for some of these places, actually understanding how much water is flowing in the system at different times of the year and how different it is from other years. One thing that really strikes me about this is just how crazy amount of water you can get in some years compared to what is the median of all years. So these are extreme rain events that are occurring over this 1916 to 2015 time period that are really like driving these incredibly high rain events and pulling this median up, by the way, as well. So there's definitely more rain in the spring and in the fall and winter in the Northeast. And then there's a lot of information about dams. Some of it is available through places like the Nature Conservancy Connectivity data portal, but there's also the Federal Energy Regulatory Commission, FERC, dam database, as well as a number of databases that we've actually put together looking at historical dams as well. So understanding where these things are is going to help you understand and interpret what fish you're getting there. It might help you understand why or why not why there are not any anadromous fish, for example, or why it seems like you have a very low biodiversity because there's not fish are not able to get into the section of river that you're interested in. And I'm not asking for you to do this for your projects. I'm just informing you about this, so when you go to your next classes, you are armed with this information, and you know when you go to do your GIS class or your fisheries class or whatever, that there's all this information out there. And when you're developing your projects, you can really depend on this. So the last thing I was going to say was about GIS products, and this is an old one that I used many years ago, which I just easy for me to put together, showing the depth profiles of the Gulf of Maine region, with some, talking about how it was flooded and glaciated back in the past, and just understanding this history of sea level rise and of glaciations and how it has effectively shaped your area in terms of the geomorphology will also help you understand why there's a bunch of sediment, for example, in a location. So all of this just helps you understand what's going on. And it also led me to really try and understand the historical perspective for these locations. So this is just one of my favorite papers by Leutz and Molesky, 2004, looking at how things had changed over time. And there's just, the first thing you notice is that we don't have sea mink anymore, neither passenger pigeons nor great auks. But the sea mink was probably really important ecologically for Northeast ecosystems, as were probably walruses, which were extirpated really early on once their Sable Island site had been they were basically wiped out there by ivory traders. But those losses have had dramatic impacts, as have the addition of a number of species, including green crabs. They created this really nice timeline sort of showing the series of changes that occurred in this ecosystem and how we arrived to where we are today. And whenever you undertake your own studies in the ocean or in rivers, understanding this history is an important part of understanding what you see today, okay? And so I'm just going to focus on the cod fishery really quickly, just to sort of give you a feeling of that, and I'll shift to river herring at some point in this talk, I think. So we've been fishing cod for a long time. I mean, a long time. And it initially was one of the only fish you could dry and ship really easily. So it was really highly sought after. Nobody would do this anymore, by the way. But these boats used to be deployed from these schooners. They would go in hand line on George's Bank, which can be actually quite shallow in places. And then they would bring their catch back to the schooner. We caught a lot of fish this way. In fact, probably more than we catch today. Salmon were also really abundant in the area, and these are weirs, and I actually show this just because it's a type of gear that we haven't really talked about very much, but it's a very effective type of gear. So all of these dots, all these little lines here along, this is going into the Penobscot River in Maine, all these little dots, this is this island here, all of these are weirs, and each of these weirs looks something like this, like hook weir or these up and down salmon weirs, all constructed of brush mostly, some particularly strange. The idea here being that the fish would swim along, hit this line, then either swim towards the coast and be like, what? And then swim back out, and then eventually along this line, and then get trapped slowly, much the same as our traps that we use, get trapped in this thing, and then harvested from there. Another one. They caught a lot of salmon. You look at the numbers here. Near 1,000, 4,000 total in 1895, and 6,000 total in 1896 in the Penobscot River. And these are the average weights based on the pounds and the number of fish. So these are big, heavy, 12 to 16 pound salmon returning in relatively large numbers. I mean, there had already been a lot of damming in the Penobscot River watershed by this time, but there was still a large number of salmon returning. Not so much anymore. But understanding these histories of both fishing with weirs for salmon, fishing with weirs for sardines can help you get information about what used to be there and how the system has changed to inform sort of your thinking as you're developing your own studies. And we depended a lot on a lot of smaller fish as well. It's not just river herring, not just salmon. These river herring, for example, were large fisheries. Often we're trying to like, why is this important? And the reason is because there are a lot of things that we're trying to accomplish now. So ecosystem-based management or ecosystem-based fisheries management, climate-ready fisheries are sort of in this whole realm of this new wave that we're trying to accommodate and understand all these connecting species that then allow us to make decisions sort of more flexibly and on the fly, and that's called adaptive management. And then we count on local management and area-based management to sort of like help in this process of getting towards this sort of, you know, the ideal of ecosystem-based management, which on its face is actually very, very challenging to arrive at. And if you're interested in that, you just have to take fisheries, advanced fisheries class, not fish ID and sampling. This a little bit dated now from 2001, showing all of the closed areas or marine protected areas in the Gulf of Maine region. There are a few more now, but not that many more. And so it's just, so when you do this, you're basically saying you can fish here and you can't fish here and you can fish here and you can't fish here, and not particularly well thought out as a whole. And so one of the things I got really interested in is like, so how are fish distributed around the Gulf of Maine? It doesn't make sense to be putting these marine protected areas sort of willy-nilly everywhere that someone says is important. And the Great South Channel is important, by the way, so I'm not saying this is bad. I'm just saying, you know, I was curious about it at the time. So my work started really close to shore and sort of started with tide pools. And I think I mentioned that really early on. And so I started working on a set of tide pools, and you can sort of see them all around here. Each of these tide pools, each of these locations usually had 2 or 3 tide pools in it. And one of the things I got really interested in was how these rocky Bass Harbor Lighthouse pools really differed from these more these smaller more marshy habitats. And in fact, the paper that I wrote based on this really just designates these sort of rocky intertidal as quite distinct and different from these more marshy leeward island sites. And that can be both in a huge island, but it can also be within a really small area as the fish community's structure just really changes dramatically from these rocky outcrops. And I'm not going to speak much more about that here. Did a lot of work in estuaries there, and one of the things I really realized as I did this work, although never published on it, was that the location of these culverts or links between the fresh water to the ocean relative to the tide really dictated what happened. So this is Bass Harbor. I kept this in there just even though, so this is me on July 4th, actually, probably 2003. And it was one of those anomalously warm July 4th. I think it was almost 100 degrees in Acadia. And I do believe that my current wife was with me on this day, but please nobody say that. I think she took these photos though. And I was just like, please don't take a photo of me right now. I am totally wiped. But she did. So I wanted to show this for a couple of reasons. First of all, because you get to see me with not white hairs on my beard. But also I was doing a survey using minnow traps that were attached to cinder blocks located throughout the estuary. And so, you know, really effective at catching certain species. And I really enjoyed collecting this data. And I had little temperature loggers in each one of these traps. I could deploy them either by foot during low tide, or I would go out and collect them with the canoe, just depending on the location. And I did it at a number of sites. So here's Seal Cove, which has a road crossing right here. Here's Soame Sound, which has a road crossing right here and a rather extensive marsh system. Mosquito Cove, which well named. Another pretty far out, like in the ocean side, with an extensive salt marsh. And then Northeast Creek, which was kind of the opposite. This is actually quite elevated and only receives inundations of salt water on spring tides. So generally fresh water, but just a very, very different system. And so what I did was, well, this is sort of it used to be a really long tour of this location, but I've cut it down quite substantially. So here's Bass Harbor. The last picture you saw was at the mouth here, and sort of showing this is one of those sites which gets a lot of salt water, floods into it, and it floods all the way up into the headwaters. So here are my two undergraduate students at the time, Johnny Spears and Natasha. Johnny was a really good friend of mine, apart from the fact that he worked with me for a year, and we've remained really good friends. He lives in Seattle now, red-haired guy, and we affectionately called him Johnny Lobster, because one day while we were out doing this, not on this day because it looks like it's about to start snowing, we went out in the sun, and at some point I was like, dude, do you have a hat or anything? And he's like, no, I left it all at home. I was like, we should go back, you're gonna get burned. And he ended up like the sheet of red. So he got affectionately named Johnny Lobster, and he's never let it down since, because a number of people call him that. I couldn't find the photo of him quickly. Natasha, I have some stories about her too, but I'll save those for another day. So this is what it looks like when you're looking down as the tide leaves, and actually behind us is this image. So when the water goes down on the other side of that structure, this is actually salt water that's flooding back out towards the ocean side. So really dynamic process, and I actually have seen I've seen squid coming in underneath this culvert or this bridge to feed in this estuary, using some of those viewing windows that you can look underwater. You'd also get mackerel and striped bass coming in here to feed on the estuarine fish as well. There's also all these salt marsh pans living, sort of on top of the marsh. A lot of mummy chugs and silver side sometimes, but mostly just mummy chugs in these. And they would get out of them only on the highest of highest tides. Just Acadia was always just such a beautiful place to work, and this is one of my favorite stretches of Acadia. So you go up Bass Harbor Marsh. Just we've caught huge numbers of alewives and other things in this creek until you get to this beaver dam. And this is one of those really neat spots where you could see crabs right next to frogs. I was like, that is not normal. But it was just that location where if any salt water was getting up here, it was right along the bottom and not on the surface. And if you go up, it just sort of kept on going into a very boggy marsh. So I put temperature loggers in a lot of locations to sort of track how the temperature was changing. And this is just to emphasize that some of these locations have really substantial temperature changes as the water warms with the fresh water warms and gets solar radiation on it. And then very quickly the tide floods in, and all of a sudden the cold ocean water comes in and drives the temperature down. Even better example at Northeast Creek. Here's one where the cold water tides are coming in and just really cooling that water off a lot. So mummy chugs are surviving this. They're surviving temperature changes from 30 to 10 degrees and just kind of being like, all right, whatever. But that's why they're one of the only species that lives there, is because they're the ones that can deal with it. So ultimately, like where you were just determined what you saw, with basically this group of species being found sort of in the estuaries, right up into the estuaries, and these fish being found sort of more further out and towards the mouth of the estuary. These are sticklebacks. The top one is a 3-spine, and the bottom one on the right is a 9-spine stickleback. And I don't know why that pipefish is there, except I think that I just was showing that they're all related. Love the sticklebacks. I was actually really hoping I'd catch one at some point and have it as your ID. There's still time, right? So I was really interested in sort of nearshore patterns as well. So here's some of the sites that I used for this study. And I bring this up because the island I went to in Maine a couple of weeks ago is actually this island. And I remember very fondly being out and sampling there. And I think we only really caught lumpfish, if I remember correctly. I didn't look at the data. But we did a lot of sampling all around that region, sort of trying to understand how the locations associated with the amount of wind energy they got. And I just calculated this based on how much available wind could come in to those locations. Then I went out, don't try this at home. And I deployed seines Now I'm in a happy mood, apparently, showing off really nice GPS gear that allowed me actually to show the exact track of, of me laying down a beach stain. So in this case, here's that line going out. So this is the same one that I showed you with Doug and Matt way earlier in the class. So what we're doing is backing, this is backing out, letting that line out, and then the net starts coming out. We deploy the net, and I'd back up and then come in forward and drop the line off. And then two people would pull that net in. We'd look happy like I did in the previous image, like that guy. And then I was really interested in associating what we caught to the location and to the regional patterns. One thing that really occurred to me though is that Mount Desert Island is really like in this Eastern Maine coastal current. And I'm not going to show you all the data that supports this, but the Eastern Maine coastal current is one of the places where there's quite an abundance still of Atlantic herring. And so we would get these pulses of young Atlantic herring coming in to use these estuaries, in particular that one that was out on Schoodic Peninsula, which is right here. We would often see huge schools of Atlantic herring coming into those locations. And you can see that they just really change. So I have a couple of different, these are 2 different years. One year, you sort of, and I think these are the same dates in 2 different years. Just sort of showing how one year can be very different from the next, and you get these patterns of really strong intensification of this Eastern Maine coastal current, and then other years it's a little bit weaker. Another couple of things you can see, this cooling here around Georges Bank is because of tidal stress that is actually mixing the water and getting cold bottom waters into the surface. And the reason why it looks really warm here is because the surface waters are trapping the heat, and there's a strong thermocline and actually cold waters at the bottom. It's actually colder at the bottom in Western Gulf of Maine compared to Eastern Gulf of Maine. It just looks cold here because the water's being mixed with strong tidal action coming out of the Bay of Fundy, being mixed top to bottom. So just really interesting area, and understanding the oceanography, putting temperature loggers in the water and capturing these plumes of cold water coming in really helped me to understand what was going on and why I would see some fish in some locations and not in others. So I then stepped out and started using some trawl survey data. So this is the Maine, New Hampshire trawl survey. I sort of divided it out into east and west initially, thinking about both benthic vertebrates, invertebrates, and pelagic species. I'm not going to get into all of the details, but I did want to take this opportunity to just mention really quickly that any trawl survey will be based around strata. Okay, and I've mentioned this one time in this class, but in this case, what they have is 5 regions which form a sort of longitudinal strata. Okay, and then they have a depth strata on top of that. So they have 4 depth strata, and I don't remember what they are exactly for the Maine New Hampshire trawl survey, but they're probably 0 to 10 meters, 10 to 20 meters, 30 to 50 meters, and over 100 meters, all within state waters. So this doesn't go all the way into the Gulf of Maine. So each of these surveys is slightly different. And the reason why you do this is so that you don't have a random survey that just throws all of the data points right around Portland. This will ensure that you get at least a couple of toes in each of the strata. So you both have a random aspect that randomly puts those somewhere. And then also you have this strata that allow those to be spread out. So you're not getting them all in one location. So what I did was, this is the boat they use, the Robert Michael. This is very typical of these trawl surveys. Now that I've been on a bunch, this comes out of the water, essentially this trawl, and you dump all the fish on a table. They're all going to be dead except for the really big. So the fish that would survive when we did them were sharks, which would scare the hell out of you. Big stingrays, which would also scare the hell out of you. And especially once their tails started whipping around if nobody grabbed them. Some of the big skates would survive. Some of the dogfish, big striped bass, big bluefish, any of the really big fish would survive, unless of course there was a giant torpedo ray in there that was electrocuting everybody, and then you knew that because the whole pile of fish would like sort of jump off the table, and you just kind of like let the torpedo ray discharge all of its electricity and then go in there and sort everything. So most of the stuff is dead. You try and keep some of that stuff for stomach contents and the things we'll talk about next week. And all of these trawl surveys can be, you can make requests for them to save those fish. And this is the only year in many, many, many years that many of these surveys didn't go out. Another reason why this pandemic's been a pain in the butt. Not that we need another reason, right? I just wanted to show this because I really like this image, even though it's not going to work. There we go. So this is from SMAS, which is down in our New Bedford. I tried to get just some straight DMF trawl data, but they didn't get it for me. It stays, and this is, thanks. This actually stays very empty for a little while. And this is pretty typical of trawls. Like you can do a tow and like not catch very much. But what you'll see, if I hopefully don't, is then they must have gone through some kind of structure on the bottom and they start seeing fish. And you guys should be able to at least, maybe not identify these to species, but recognize that these are flatfish going through. These are probably silver hakes is my guess, just based on how abundant they are. Most of these look like winter flounder. Here's a skate. That's like not a winter flounder. That's probably either a yellowtail or a window pane. More skates, and then you start seeing some scallops flying by. And I think eventually we see some haddock and cod come into this. Anyway, I'll post this up. There's some kind of hake. Oh, and there's a dogfish. So one of the things that is going on in this work by SMAS is they're actually looking at using these video cameras, and I know Kevin Stokesbury has way better image photos of this, but they actually are trying to automate and have the net be able to ID or corral these fish, and then they have a video camera that's taking these images, and then they run it through a computer algorithm that detects the number and species of fish that are going through, so that we don't have to catch them. One thing you'll notice is this is tow 15 closed. They actually have the net closed, and they're going to enumerate all of the fish eventually once they get on deck. And then they run this with the net open. So they're just seeing, that's a bigger flounder. So I'm going to guess that's probably a large summer flounder. So they are trying to get to the point where they are going to be able to do this automatically and not have to catch any of the fish, which would obviously be great. You're going to always need to catch some fish because we do need information that we'll talk about next week in the post